Hi, my name is Christopher Kirkwood.
I'm Shania Farahi.
I'm Griffin Edward.
And I'm Joseph Piovazan.
We also have a fifth member, but she was not able to make it today. Her name is Vasal Farahi. We are the Redworm Composting Group. We would like to thank our teacher for making this all possible for us. Our group is sending redworms into space with the aim of gaining knowledge that could help with future space travel and missions to Mars. On August 10, 2015, the astronauts aboard the ISS harvested and ate romaine lettuce that they planted in space in July. Redworms eat food scraps and then excrete good nutrients, helping newer plants grow. This is called composting. With our worms in space, we'll be able to determine how redworms grow and function in microgravity. Astronauts lose their bone and muscle mass while in microgravity. Redworms have no bones, but they do use their muscles to burrow. So we want to know whether or not redworms would have enough muscle mass to burrow in microgravity. Redworms are also a very important part of composting toilets. A composting toilet is a regular toilet, except that after the urine and feces go into the toilet, a mechanism in the toilet separates them and the feces go into a giant metal canister. Inside the canister are redworms. The redworms then turn the waste into fertile soil that could be used to grow food. If the redworms can grow in space, we could potentially have a fully functioning space garden with soil composted by the worms from the fecal waste produced by the astronauts. Having gardens would save the cost of transporting food into space because astronauts could grow their own food. As well, redworms could eliminate the cost of waste capsules that are currently being used to dispose of human waste.
When we first started, we wanted to send up a spider. What we wanted to see was if spiders could spin webs and make silk in microgravity. We did some research on it before realizing that there wouldn't be enough room in the FME, that we would need to find non-venomous spiders. After that, we scrapped that idea and decided to send redworms into space. We chose redworms because the worms can compost waste and turn it into fertile soil for gardens. With gardens, astronauts can grow their own food. Another reason we wanted to send worms is we thought that maybe if they can hatch and grow properly, we might be able to send other small animals. Spiders would be no use to the astronauts aboard the ISS, but worms would be. SSEP was a massive undertaking, and to come up with an insightful proposal took a community. When we were generating ideas for our project, grade 12s from our local high school came down and sat down with us to discuss the possibilities. Engineers from local businesses such as the You Are the Cast, who make cameras for the ISS, Skyped in with us and helped us to understand what science in space looks like. Professors from local universities took the time to visit our schools and extend our thinking. After we refined our ideas, parents and community volunteers took time out of their busy work schedules to help us edit our proposals.
Since we will be looking at the worms under a microscope when they return, we will look at the anatomy and biology of red wigglers and their cocoons' weight. The cocoons can take 3 weeks to 5 months to hatch, so it could potentially lengthen the experiment by almost a half year. We want to freeze the redworms in place so when the astronauts get up to the ISS, they will unfreeze the worms and they are ready to grow up there rather than down here on Earth. After the experiment is done, we will see if there are any changes in the worm's muscle, tissue, mass, etc. by dissecting the worms from the ISS and from the ground experiments after the entire thing is done. We don't know what the exact consequences are going to be if something goes wrong, but it will be worth a shot to see what happens. We use the Type 3 FME. In Volume 1, we put redworm cocoons that we got from our classroom's worm bin. In Volume 2, we put 4.4 milliliters of moist dirt. Mixed in with the dirt, we put some leaves, and that is which the worms get the nutrients. And lastly, in Volume 3, we put 2 milliliters of 10% neutral formalin or 4% neutral formaldehyde. Neutral formalin, or NBF, is a fixative that will freeze the redworms, ensuring that their growth only occurred in microgravity. Our experiment will be initiated in the first week of arrival at the International Space Station by having our astronaut release Clamp A and shake the mini-lab gently for 10 seconds. The procedure will mix the one redworm cocoon with the 4.4 milliliters of moist dirt to help the worms survive in the absence of gravity conditions. The moist dirt will be made up of dry dirt and water. An astronaut will release Clamp B and shake the mini-lab vigorously for 20 seconds, freezing the redworms in place. Freezing the redworms with the fixative will undoubtedly make sure those worms only grow on the ISS. While the FME is microgravity, we conducted the same experiment here on Earth but did it a little bit earlier. To eliminate the number of variables, we will have the red worms in the exact same living conditions, except we left out microgravity because of obvious reasons. The red worms will be in a tube similar to the FME microlab, and there will be the same amount of red worms, dirt, and the fixative will be used as well.
Our ground experiments. To test the ideal living conditions of the earthworms, we ran tests at our school. The tests include different amounts of dirt, water, and types of food. For our first experiment, we tested the amount of water. We started with 2 milliliters of water, then we went to 4, 6, 8, then 10. In each test tube, we had 6 milliliters of dirt and 4 red worm cocoons. In the second experiment, we tested the amounts of dirt starting with 3.5 milliliters and increased it by 1 milliliter each time. The fifth test tube had 7.5 milliliters of dirt. We then added 4 milliliters of water and 2 worm cocoons. In the third experiment, we had 10 milliliters of dirt and 2 cocoons in each tube with 4 milliliters of water. We tested different types of food, dirt, and we had carrot, celery, apple, orange peel, and bits of leaf.
Next steps. When the worms come back from the ISS, we'll compare the experiment done in space to the one done here on Earth. We will carefully look at the dirt for any wet red worms. If they haven't hatched, we'll look closely at the cocoons under a microscope. We'll compare the cocoons from space to the ones done on Earth, and look for any explanation of why they didn't hatch. If there are worms that have hatched, we will take a look at them under a microscope. We will look for any changes in growth. For example, we are going to look for any changes in physical features such as missing ligaments. We will then weigh them to see if there has been any weight change. Finally, we will dissect the worms and look for any changes or missing organs.
And thank you to our sponsors for making this whole trip possible.
Thank you.